Okay, this is the last lecture for Chapter 3, and it's going to be probably the shortest lecture, and in my opinion, the easiest and funnest part of the chapter. So now we're going to talk about pedigrees. Now, we can't experiment on humans, right? We can't put a man and a woman in a lab and have her have 20 children and do crosses of particular traits and things like that and look at the progeny. And typically in nature, we don't have large numbers of children. Like I said, unless your last name is Duggar. I think she's up to 19 kids. So we have to be creative, study inheritance patterns in people. And so we use pedigrees. So we just have to look at people's histories in families. And in some cases, if it's pretty straightforward, we can actually determine certain things about traits. We can figure out whether or not that trait is dominant or recessive. If the gene that carries that trait is autosomal, meaning it's present on chromosomes other than the sex chromosomes, or if it's X-linked, which would mean that it's present on the X chromosome, which that'll be a big part of Chapter 4. So what we're going to really talk about today are dominant and recessive traits on autosomes. All right, so just go over a little bit about pedigrees, what they look like and what the symbols mean. Usually the affected individuals, so the people that are displaying whatever the trait is that you're studying, are going to be shaded in. You've got circles from females, squares from males, a line between a circle and a female and a male means that they're the parents. If you have two lines between them, it means they're consanguineous, which means that they are related by blood, which, doesn't happen so much now, but when you look at old histories, especially like in royal families and stuff, you're going to see a lot of that. So if you have twins, then that's going to look different. Here you can see the twins look different than just individual siblings. So these are siblings, and this is called a sibling, I think, right here, that joins all the siblings together in a family. Identical twins look even different. They look like twins, but they have an additional line there to point out that they're identical. Sometimes for shorthand, if you have families with a lot of people, they'll just put a number in there that says, it's multiple individuals that are unaffected. The proband is the actual target, the reason that the pedigree is being studied in the first place. So you've got this affected individual, and you want to know where they got this trait from. So then you start to study the family, and you go up and down and sideways, looking at all the family members. If it has a line crossed through through the symbol, then that means they're dead. And if it has a dot, then that means it's a heterozygous carrier. And sometimes they'll let you know that, and sometimes they won't. And then generations are marked with these symbols. And then you have Roman numerals give you generations, and then regular numbers are the number of children in the family. Okay. So enough about the symbols. All right, so the first kind of trait we're going to talk about is an autosomal recessive condition. And albinism is a good example of that. So this is people that, for whatever reason, it's a loss of function of their genes that code for the protein for the production of melanin, which is a skin pigment. So they don't have any pigment in their skin. So they're very pale. They have no color to their hair. Sometimes their eyes are actually red or very light blue. And of course, like I said, this condition is a recessive condition. So you have to have two mutated alleles in order to have the trait. And it's probably arose from some kind of mutation in the gene that made that gene no longer functional. And again, the individual is going to be homozygous recessive. All right, so here's an example of a autosomal recessive pedigree. And so you can see here, if you have, here you've got a couple here that doesn't have any children with the trait. But clearly, one of their children is a carrier. here. So this child is a carrier for the trait. So really we should put a dot in the center, right? Because, and then this person has to be a carrier. And we know where this person got his allele. It came from this father. So this father was affected. The mother was normal. More than likely she wasn't affected. If she had been affected, then they would have had probably at least one of these three children would have also been affected, because it would be a much higher incident of chance of them getting two alleles. So the fact that none of these children have it, it's unlikely that the mother was even a carrier. But these two obviously were carriers. They're heterozygous because they produced a child, actually two children, that have the trait. Like I said, neither one of these, I'm sorry, one of these at least is a carrier, and we don't know which one it is. But it's unlikely that both of them are carriers. Otherwise, one of these four kids more than likely would have had it. It's not for sure, like I said, because when you only have like four kids, you know, it changes the probabilities. But more than likely, you would have seen at least one child affected. So it's probably the case that only one of these was the carrier, and we can't tell from this one which one it was. So we know that either one of these was a heterozygote carrier. Notice here you've got affected in generation 1, no one's affected in generation 2, but then generation 3 some more affected ones pop up. And that's because recessive traits typically skip generations. And because it's autosomal, because it doesn't have anything to do with the X chromosome, these traits will appear equally in both sexes. All right, so just some hallmarks of recessive inheritance patterns. Usually the mutation results in a loss of function. So like, for example, with albinism, the cells can't produce melanin anymore. If you're heterozygous and you have one good gene and one bad gene, usually the one good gene is enough that you'll appear normal. It'll produce enough gene product that you won't exhibit the trait. Now, parents of an affected individual are usually normal heterozygous carriers. You know, and then especially when it comes to disease states like sickle cell anemia, and which is going to be a big part of your homework this week, or cystic fibrosis, you know, something like that that's very debilitating. Usually the parents are normal carriers instead of actually being affected. And so if you have these two heterozygous carriers, you have about a 25% risk of having a child with the trait. And like I said before, the trait usually skips generations. You know, if you have two affected individuals by chance and they actually have offspring, then of course all the children will be affected, because that'll be the only alleles that they'll be able to inherit from both parents. So some conditions that are recessive: cystic fibrosis, like I said, sickle cell anemia, that's a big part of your homework, and the albinism example that we just saw. Okay, so brachydactyly, this is a dominant trait. This is an autosomal dominant trait. It is characterized by having abnormally short fingers or toes. Just joking, maybe Donald Trump suffers from this. And the reason I thought about this was because of like Jimmy Fallon and some of those other night hosts, comedians, talking about Donald Trump being so upset because somebody said he had tiny hands. But tiny hands is not the same thing as unusually short fingers. So, but anyway, so that brachydactyly is a very common dominant trait. This guy Farabee, he observed, he looked at this condition in families and tried to figure out how it was passed. All right, so this is going to be different from albinism because this is a dominant trait versus a recessive trait. So in albinism, we saw that usually with these recessive traits, there's some kind of loss of function. In brachydactyly, usually the gene gets mutated and performs a different function. So whatever the gene is, it's supposed to make fingers the proper length. It's doing something different now, and so not all the fingers are the same size. So this is a dominant trait. If it's a dominant trait, that means you only have one gene that has to be affected for the condition to be present. So this is an example of an autosomal dominant pedigree. Now notice some things. Notice here the dominant trait is almost in every single generation. So, well, in this one it is in every generation, and that's typical with a dominant trait. You have here generation 1, and then we got a couple people in generation 2, and then we got some more people in generation 3. So every generation displays the trait. In order for you to be affected, or a child to be affected, one of the parents has to be affected, because they have to be able to pass down that trait. And if they have the trait, then they're affected, because you only have to have one copy of the allele to be affected. And the dominant autosomal traits appear equally in both sexes, again, because it's autosomal. If it was on the X chromosome, it would be a different story, which we'll see in the next chapter. And typically about 50% of the children of an affected person will be affected, because they have basically a 50-50 shot of inheriting that allele that produces the trait. But you can see, again, because of chance, that's not always the case, right? And this time you only got 1 out of 4 when it should be 50%. And it is very rare for someone to be homozygous dominant. But if they were, then all of their children would be affected. All right, so hallmarks of dominant inheritance. These are gain-of-function mutations. You only have to have one faulty gene to show the trait to be affected. If you inherit the gene, then one of your parents must have been affected as well. If only one parent is affected, then 50% of the children will be affected. If by chance there's 2 heterozygotes, there's 2 affected individuals, then 3 out of 4 of their kids will be affected. And if one parent is homozygous for the trait, which, like I said, is rare, but is possible, then all the children will be affected. So some examples include brachydactyly, which we already talked about, dwarfism, and Marfan syndrome, which we'll talk about more. I think in chapter 4 we talk about Marfan syndrome. It's believed that possibly Lincoln had Marfan syndrome. All right, so now the last thing we're going to talk about is just some molecular explanation. So, we know that there are 2 alternate forms of the peas as far as their shape is concerned. You have round and wrinkled, and we know that they're controlled by genes. But what exactly is going on with these genes, and how do we end up with these wrinkled peas? Well, it turns out there's a gene called the SBEI gene, and it's involved in, it's an enzyme that's involved in creating branched structures of starch. So as the seed is making glucose and then storing that glucose as starch, the enzyme catalyzes the formation of the branched starch, so that you have all these different branches. In wrinkled peas, this gene is non-functional, so you don't have a lot of branched starch structures. Instead, you just have really small sugar molecules of just glucose and fructose, which is sucrose, and then that causes water to build up, which increases the osmotic pressure. And then the pressure gets to the point where the seed loses that water, and it gives it a wrinkled appearance at maturity. And so that's why those seeds are wrinkled, which makes me think they would probably be sweeter, but because of the sucrose. But I don't know if wrinkled peas are sweeter than brown peas. So what gets even more interesting is when they actually analyze this mutant gene, they find that it has something in it called a transposable element. So this is just a sequence of DNA that's kind of, it's a foreign piece of DNA that's kind of wedged itself into this gene and interrupted the gene sequence so that it's no longer functional. And these transposable elements are found all over the genomes of all kinds of organisms. Some are thought to be viral in nature as far as where they came from, but I think later in the semester we'll talk about transposable elements. There's a lot that's still unknown about them, but they do have a tendency to wreak havoc in the genome, and in a lot of cases they'll cause some kind of loss of function of a gene. Okay, so the last one we're going to talk about is Tay-Sachs disease. Tay-Sachs disease is a autosomal recessive trait. It in children that inherit have parents that are carriers and they inherit two faulty genes. They will appear normal when they're born. At about 6 months, though, they'll start to have progressive loss of mental and physical abilities. They become blind, deaf, mentally retarded, paralyzed. Typically, they die before the age of 5. And this results from a loss of activity of the enzyme called hexosaminidase A, or HEX-A. This is an enzyme that breaks down a molecule called ganglioside M2, which is a lipid component of nerve cell membranes. And so these gangliosides, because they're not being broken down by this enzyme, they'll accumulate within the neurons, and eventually they'll kill the neurons. So it causes deterioration of the brain and the nervous system. And it's basically just their brain and their nervous system breaking down over time. And so that's why they don't live very long at all. Now, if you have one copy of the functional gene, then you appear normal and you don't have the disease and you can live a long, healthy life. So apparently, just one copy of the gene makes enough of this enzyme to prevent the buildup of these gangliosides. All right, so every trait that's out there, whether it's sickle cell anemia, which you'll learn more about the molecular basis of that in your homework assignment this week, or for cystic fibrosis or familial hypercholesterolemia, all these different diseases that are inherited diseases have some kind of molecular explanation. So, all right, with that, that's the end of chapter 3. We're done. You can finish up this lesson by answering the review questions. Make sure you do the homework, and there'll be a quiz on this chapter at the end of the week. And like I said, review, because the first test will be on chapters 1 through 3, and that will be due next week. So with that, I'm going to end this lesson.